Hello everyone. Welcome to the online classes of biology for class 12th. Today we will discuss about the following subtopics: fertilization, implantation and placenta formation. All these topics are from chapter 3, human reproduction. Now about the learning objectives of this session. The learning objectives are fertilization, site of fertilization, monospermy, implantation, zygote development, placenta formation and its role. That means after completion of this chapter, you will be able to relate with all these terms as well as understand it better. Now the first topic for the session is fertilization. During copulation, the semen is released into female genital tract. This is called insemination. The motile sperms move through the cervix, enter the uterus and reach the ampullary isthmic junction of fallopian tube. Fallopian tube is also termed as oviduct. The fusion of male and female gamete is called fertilization. Fertilization can only occur if ovum and sperm are transported simultaneously to the ampullary isthmic junction, which is also called as the site of fertilization. This is the reason why not all copulation lead to fertilization and pregnancy. The secondary oocyte released from the ovary also reaches the ampullary isthmic junction where fertilization takes place. As the picture is also showing that there are many sperms which are moving towards the secondary oocyte and only one of the sperm will enter it by penetrating the zona pellucida. The sperm which will come in contact with zona pellucida will induce certain changes in this membrane. These chemical changes are a series of cortical reactions which converts the zona pellucida into fertilization membrane. This membrane acts as a blockage for the entry of additional sperms. It is called as monospermy. Now, you all are able to answer the question that why monospermy is found in human beings. The secretion of acrosome help sperms to enter into the egg cytoplasm, that is the secondary oocyte, through penetrating zona pellucida and vitelline membrane. Acrosome is formed by Golgi body and it contains sperm lysins hyaluronidase which help in penetration through the layers of egg. This induces second meiotic division of secondary oocyte to form an ovum and second polar body. The haploid nuclei of ovum and sperm fuse to form diploid zygote. Even before the nucleus of the ovum is organized, it fuses with the sperm nucleus to form a zygote. Now the second topic of the day is development of zygote. The picture is showing that there is secondary oocyte, that is A, and many sperms coming towards the ovum, and one of the sperm is able to penetrate the layers and fuse with the ovum. Here, after the formation of zygote, it undergoes successive mitotic divisions called cleavage, and the daughter cells are called as blastomeres. All these are the stages of developing embryo. The mitotic division starts at the zygote, moves through the isthmus towards the uterus, and forms 2, 4, 8, 16 daughter cells. These cells are called as blastomeres, and the embryo at 8 to 16 celled stage is called morula. Morula continues division to form 32 celled stage called blastocyst. This stage is called blastocyst because it encloses a cavity called as blastocele. The blastomeres in blastocyst arranged into an outer layer which is called as trophoblast or trophoectoderm, and a inner group of cells called inner cell mass or embryoblast. The fate of trophoblast is to form 4 extra embryonic membranes, and the embryoblast will form the embryo. The blastocyst get attached to the innermost layer of uterus, which is called as endometrium. After attachment, uterine cells divide rapidly and cover the blastocyst. As a result, the blastocyst get embedded in the endometrium, and this is called as implantation. Now, next topic is placenta formation. The picture is also showing the connection between the developing embryo and mother's uterine tissue. After implantation, finger-like projections called as chorionic villi appear on the trophoblast, which is surrounded by the uterine tissue and maternal blood. The chorionic villi and the uterine tissue become interdigitate with each other to form placenta. Placenta is a structural and functional unit, or we can also say that it is a vital link between fetus and the maternal body. Placenta is connected to the embryo by an umbilical cord. It transports substances to and fro from the embryo. The exchange of substances between the mother and the developing fetus takes place through the placenta. Now the functions of placenta. It facilitates support of nutrients and oxygen to the fetus. It also helps in removal of carbon dioxide and other harmful substances or excretory products from the fetus. It acts as an endocrine tissue and secretes the following hormones, that is HCG, human chorionic gonadotropins, HPL, human placental lactogen, progesterone and estrogen. Now we have completed all the subtopics for this day. Hope you people have enjoyed the class. Take care, stay safe. Thank you.